We present Nanomatrix, a method for interactive construction and rendering of extremely large molecular scenes, capable of representing multiple biological cells at atomistic detail. Nanomatrix is a view-dependent procedural approach that generates geometry at atomistic detail of the visible portions of the view on demand on the fly. We define a scene as a virtual grid of cells. Atomistic details are generated only inside the active cells surrounding the camera. For populating molecular instances, we procedurally generate 3D geometry patches using a rule-based method. Within this patch, 4 subpatches are determined and Wong tiles out of them are generated. Possible collisions are handled. Afterwards, geometric Wong tiles can be combined together by matching colors to create a seamless non-repetitive patch denoted as the tiling recipe. From geometric Wong tiles, we generate corresponding 2D textures. These will be used for texture sampling when rendering meshes. We introduce a view-guided construction algorithm that uses the seamless tiling recipe to populate the scene on the fly. To populate one triangle of a membrane, amount of tiles needed is computed. Then, instances outside the triangle and the currently processed cell are discarded. This is how the mapping looks like when applied to more complicated mesh. Interior of structures is filled by predefined 3D box tiles. Molecular instances are placed only inside based on in-out test with respect to the mesh. The algorithm is not restricted by the size of the mesh. Wong tiling can be recomputed on the fly, resulting in the different variations of populated membrane. We use a parallel rendering scheme that utilizes hardware-accelerated ray tracing for molecular visualization. Details closer to the camera are rendered at the atomistic resolution, while objects further away from the camera are rendered as textures. Selecting which cells should be filled with atomistic detail can be done in several ways. Here side-by-side comparison between selecting the closest cells to the viewpoint versus selecting the ones that intersect the view frustum.